Okay. Hello, everybody. We are going to be talking about specifically this Edpuzzle. Or I keep saying Edpuzzle, this video. We're going to be talking about scientific skills and scientific processes. Um, if you are thinking about science, remember that the goal is to solve problems, right? To answer questions and solve problems. So when we think about the scientific process, it's ongoing. And scientific skills can be used in lots of different ways for lots of different reasons to solve problems throughout your life. So while you might not consider it the scientific process, it's still a process that you're going to use when answering questions in lots of different ways. So this year, we're going to be talking about science in a couple different ways. Our new Pennsylvania standards are going to be looking at core ideas, scientific and engineering practices, and cross-cutting concepts. So concepts that connect across a variety of different scientific disciplines and then also connect our units together. So these are the three lenses we're going to look at science through this year, and you'll see them in a variety of ways as we move through. So some important terms to know when you're talking about scientific process. Observations are something that you can physically see. It's something that you can observe happening or observe in your world. An inference is a guess or idea that you are basing on evidence. So, based on what you've seen in the past, you can create an inference about what's actually happening in a scenario. And then a theory is the explanation of some aspect of the natural world based on a body of facts or a body of evidence that have been confirmed through observation and experiment over and over and over again. So things that we have studied and looked at and analyzed and evaluated many, many, many times, eventually all of that information comes together to form a theory of what actually happened. So, because the scientific method is an ongoing process, you usually start at um, making observations. Like, what do you see happening in nature? What's happening in the world around you? And then the next thing we do is think of questions. So, like, why is this occurring? What is causing this pattern? What's causing this phenomenon? What's causing this um, thing that we're seeing to occur? Then you're going to formulate a hypothesis, which is like your guess about what's actually happening. So this would almost be like an inference based on what you're seeing. What do you think is happening? So that's what formulating a hypothesis is. And this is all things that you guys have learned, that you've talked about, that you've done several times in the past in your science classes. Then we're going to develop a testable prediction. So that's like your hypothesis. So if your hypothesis is correct, this is what you're going to see. Okay. Once you do that, you're going to gather your data, you're going to test those predictions, you're going to carry out an experiment. And then based on your results, you're going to refine that experiment, change it, expand it, accept your hypothesis, reject your hypothesis, and then move on. If you accept your hypothesis, though, generally you can start to develop a general theory. But again, you're going to have to go back into that process. You might not go back to the observation step. You might go back to the interesting questions, like what's something that happened you didn't expect. So these are all things that you continually do throughout the scientific process, and then we're going to work through this year. So when you look at an experiment, your experimental setup is going to start with your hypothesis. That's your guess. That's your prediction of what's going to happen. You'll need to identify your independent and dependent variables. Remember, independent variables are things that you change to test what's going to happen. So independent variable is something that you're changing in your experiment. It's going to create your experimental groups. Okay. The dependent variable is the data that you're collecting. It's responding to the changes that you've made. So the dependent variable depends on what you do. Once you identify those things, you're going to set up your procedure. You're going to set up like what you're going to do, the specific steps. Sometimes this is called a methodology. You're going to set up your procedure. You need to identify what's going to be your control group. Maybe it's the norm of what happens. Maybe it's like a baseline of what happens, but you have to identify that control group. And then you have to identify the things that are going to stay constant in all the groups, including the control group and all of your experimental groups. So that experimental setup is going to be the same all the time. That allows you to collect valid data. Now, there's two types of valid data that you're going to want to collect. Qualitative data, that is evidence, that's a description. So maybe you're looking at the number of, or sorry, not the number. Maybe you're looking at the color of flower or the color of leaves on a plant. That is qualitative, things that you can just observe. Quantitative is data or evidence that can be expressed as a number. So, for example, qualitative could be the color of the flower or the color of the leaves. Quantitative would be the number of flowers that you count or the number of plants that you're counting. So these two types of data are both very important, and it requires you to take important notes while you're doing an experiment, but you need to make sure that you are collecting as much evidence and as much data as possible in any experiment. Once you've collected all of your data, you have to determine how are you going to represent this data, because sometimes it just looks like a big old set of jumbled numbers on a chart. You're going to set up your graph. And we've talked about the graphs before. We've done a little graphing practice, but just as a reminder, this is your y-axis is going to go here. So your y-axis is here. Your y-axis generally is going to hold your dependent variable. There are some cases where you might put your dependent variable on a different place, or maybe you have multiple dependent variables. So this, for biology, we're treating this as your dependent variable is going to go on the y-axis and your independent variable is going to go on the x-axis. So the dependent variable responds and it goes on the y-axis. The independent variable is the manipulated variable, the one you're changing. It's going on the x-axis. And remember that your title is always y-axis versus x-axis. Now, I know that in other classes, in Mr. Petsko's class, he has told me that sometimes these rules aren't hard and fast. That's true. This is a good place to start if you're not sure how to set up your graph, okay? So maybe not 100% every single time, but in biology, this is how we're going to set up our graphs. Dependent variable on the y-axis, independent variable on the x-axis. Now, moving forward, we're going to start talking about ecology, and these are some of the other topics that we're going to be talking about. But as we move forward through our year in science, we're going to be consistently using that scientific method. So make sure that you understand all of these different terms, because we're going to be coming back to them over and over again as we observe and we make predictions, we ask questions. All of those steps of the scientific method are very, very important.